Good afternoon. Today I'm going to present the findings of a study that we have been doing at a coastal marsh system in southern Alabama. This was a marsh system that was inundated by oil from the Deepwater Horizon oil spill, and we are taking a look at the microbial community and how it responded to this oil. Now the blowout of the oil excuse me, make sure this works. Okay. The blowout occurred on April 20th, 2010, and as this map developed by the New York Times shows that the oil inundated the Alabama coastal system, and here it's shown in gray, on or before July 2nd. Now our system, or our study site, is about 15 miles to the west of Mobile Bay. It's along the Point of Pines Peninsula, and this is just south of Bayou La Batterie. Now this area is very important economically to the state of Alabama. It's considered the seafood capital of Alabama. It brings in several $1 million from the seafood industry as well as from tourism. Now, our site is a typical marsh system, typical of the Gulf Coast. It is inundated with juncus and spartina grass. We also have oyster beds present. On July 8th of 2010, the oil was seen at the site. It was presented as a sheen on the water itself. It also was collecting as tar balls or tar mats along the shore, and it was also collecting on the marsh grass itself. And so the marsh grass was acting like a trap for it and allowing it to go down into the sediment. We collected in two different sites at this location. We collected sediment cores within the marsh grass itself and also approximately 25 meters from shore away from the grass. We collected during the first part of June of 2010, July, September, October, and then we have continued back at this site approximately every 2 months up until the present time. Now, we measured the oil contamination at the site by two different methods. The first method I'm going to show is by total petroleum hydrocarbons, or TPH. And as this describes or explains, it is basically a total bulk measurement of total petroleum hydrocarbons. And what we found at the site was that it ranged very sporadically or very randomly throughout the system. It was very heterogeneous. It ranged from approximately below detection to up to 189 milligrams per kilogram. Now, just as a guideline, Alabama cleanup standards for TPH is 100 milligrams per kilogram. We also measured it with GC mass spec, which actually can separate the different alkanes and then quantify them. We used the Macondo wellhead oil itself as a standard, and you see that it is typical of a light Louisiana crude, and that is enriched in the lower chain or the shorter chain alkanes, C9 through C17. And when we analyzed our tar balls as well as oil from the sediment, we found a very weathered signature in that the short chains were depleted and we had primarily enriched longer chain. This is very typical of a weathered oil. And so just in a summary of this, the tar balls and the oil within the marsh at the Alabama coast was depleted or degraded compared to the original oil from the wellhead. It was depleted in the short chain alkanes, and it was enriched in the long chain alkanes. It has a pristane to phytane ratio, very low, of 0.2, and this is on a scale of 0 to 1. That is where 1 would be the original oil. The TPH concentrations were highest in the sediments amongst the grasses. Like I said before, the grasses were acting as a trap for this oil and allowing it to descend into the sediments. By October of 2010, the TPH concentrations were below detection, and just also as an aside, we measured the TPH in this past June of 2011, and we did not detect any TPH at that point. Now, just as a quick cartoon of how oil biodegradation occurs, if you have oil in the presence of a microbe, you can get the final products of carbon dioxide and water. However, for this particular biodegradation to occur, there are several factors that affect it, and these are very important for determining how enhanced this biodegradation is or how limiting it can be. And so for our analysis on the microbial determination of this sediment, we used the Berkeley Lab PhyloChip 3. This is a very unique and very powerful tool in that you can detect up to greater than 50,000 different bacteria and archaea in one test, and it's done rather quickly. And so when we analyzed our samples from this site, we find that it is very diverse microbially. There is greater than 14,000 unique microbial types found at this marsh. And this is in comparison to, say, an open ocean system where a similar test was run, and there was only approximately 950 microbial types at that site. In addition, there was 63% more hydrocarbon-degrading bacteria in those samples that contained oil versus those that did not contain oil. In addition to this analysis, we also ran a similar test by the geochip microarray analysis, which is similar in that where the phylochip measures the bacteria and the archaea that are there, the geochip measures the actual or determines the actual genes that are present in the sample. And these, if we take a look at the specific hydrocarbon-degrading genes for these sites, we see a positive correlation between the amount of oil that is in the sediment and the genes that are degrading them. So in summary, the oil contamination at our Pornapine salt marsh was detected in June, July, and September of 2010. However, after that time, in October and then also in June of this past year, there is no detection of total petroleum hydrocarbons. The phylochip as well as the geochip analysis allowed us to determine that the bacteria that were working in the sediment during these times were enriched, or the sediments were enriched in these hydrocarbon-degrading bacteria, as well as the geochip indicated that the genes that were used for breaking down these hydrocarbons were in excess. The microbial communities responded rapidly to the oil input at this site, and so this was most probably due to the fact that the factors that contribute to the degradation of oil in a sediment were optimal, such as the temperature, the pH, the nutrients. This is an area where you have runoff from the land, and so there is nitrogen and there's phosphorus there for the organisms. Also, the light crude, the type of crude that came in, as well as the fact that it did not oversaturate the system, allowed the microbial community to degrade the oil. And the most important part, I believe, is that the mixed microbial community that is present there. This is a system that is naturally affected by hydrocarbons because this is a naturally seep system in the Gulf of Mexico. So the organisms that are there that can degrade hydrocarbons are naturally there. And so when they have an input of hydrocarbons, they are able to degrade it very quickly. And so with that, we have concluded that natural attenuation at this site was a success in this particular instance. So, thank you.
Hi, thank you for your interest in our research. I don't have any slides for this fairly brief introduction of what I've been doing with the U.S. Geological Survey, looking at the ecological effects of the oil spill. There are a number of people within the U.S. Geological Survey that are doing various types of monitoring, and our work within my science center in Denver started in July of last year. The initial work was for a group of remote sensing people to look at on-ground signals for remote sensing and compare it with flyovers to understand how far the oil spill had spread. And because they were in the field doing this work, there were some advantageous samples that could be collected to look at what happens to carbon. Carbon is a major elemental composition of oil, of course, but the question that I had as a biogeochemist looking at cycles of nutrients like carbon is what exactly happens to that carbon, what form does it take, what is the biodegradation pathway of the actual molecular form of petroleum compounds. How do those compounds get partitioned into water? Do they get partitioned in the water? And then what does a dissolved component look like? Dissolved organic carbon is, by definition, the carbon that passes through a 0.45 micron filter. And it can be measured directly as a concentration of dissolved organic carbon, but DOC also fluoresces. And the fluorescence is characteristic of what the sources of the organic matter, for instance, Mississippi River organic matter, which is ultimately the source of organic matter in these marsh ecosystems, has a very distinct pattern. And the pattern that was formed by dissolved organic carbon that was taken directly offshore from the area where the oil slick was quite different than that. So the first phase of work was looking at the offshore oil slick. My colleague Bill Oram, who's in the U.S. Geological Survey in Reston, Virginia, was looking at the petroleum compounds in the unfiltered fraction. And I was working with my colleague George Aiken, who's in Boulder, Colorado, looking at the dissolved organic fraction of carbon. So the first phase was looking at the offshore materials. And then that next phase was looking at what was hitting the marshes in Louisiana. There are two areas that we looked at, also in conjunction with a remote sensing study that has been published as an open file report, to see where oil has hit the shoreline. And the two areas that were looked at were the delta area, the little nose that's a distal part where the sediment has been deposited by the Mississippi River, and then Barataria Bay, which was in the news quite a bit last summer because that was the shoreline that got hit the hardest in Louisiana. And this phase, the shoreline survey, allowed me to collect water from a number of different places along the shore, water just at the surface of the marsh that was interacting with the marsh organic matter and the organic matter derived from petroleum. And that material had a distinct fluorescence that wasn't quite like the petroleum, and it wasn't quite like the organic carbon that would come off of the Mississippi River sediments. It was somewhere in between, in both the visibly oiled sites and the sites that didn't have any apparent effect of oil. So when I say visibly oiled, in the field it was just blatantly obvious. There was a slick of kind of a caramel-colored oil that was along some shorelines, and the not visibly oiled had intact vegetation. The oiled sites tended to have the vegetation die. And we were able to do a follow-up in October, working in conjunction with our colleagues in Baton Rouge, who have a series of 6 study sites along Barataria Bay. These 6 study sites include 3 unoiled or visibly unoiled sites and 3 oiled sites. And this follow-up study has proven to be kind of interesting so far. I've only had the data, obviously, for a short period of time since the samples were collected in October. But there are distinct differences in how the dissolved organic carbon fluoresces in the oiled pore waters, so the pore waters being the subsurface solution that's moving through the soil material. And there is a greater degree of fluorescence in the sites that have been impacted by oil than the sites that have not. And in the following months, I hope to work with my colleague who is looking at the characterization of petroleum compounds to see if I can determine whether this fluorescence is caused by natural organic matter that's being formed as vegetation is decaying and dying, or if it is a result of continuously biodegrading petroleum compounds. So that, in a nutshell, is what I'll be talking about tomorrow. Thank you.
Excellent. We'll now start the question and answer portion of the press conference. The first one I'll take from the webcast. Seth Bourne, speaking at AP, this question from Melanie Beasley. How can you determine the difference between natural degradation of oil by microbes and the degradation from added dispersants?
From the analysis that we have conducted, we cannot determine that exactly. However, the dispersants, from what I understand, were applied the 6 months prior during at the actual wellhead. Now, how that dispersant arrived at Alabama approximately 6 weeks later, I cannot say. We cannot determine from the actual tar balls, or at least from the analysis that we have done.
Lizzie Grossman, freelance, and I was going to ask a question kind of along those same lines, but a little bit differently for either or both of you, including Joanne. How, from the work you were doing, would you determine the petroleum products that were involved in the dispersants versus the ones that were in the oil, since there was so much mixing and that oil was so terribly dispersed and diffuse?
It could be a challenge to be able to tell the difference between the degradation compounds of the dispersant and the degradation compounds of the petroleum products themselves. The dispersant technically does have a lot of proprietary ingredients, but fundamentally the molecular structure of the petroleum could be a lot more complex. We don't really have separate samples of dispersant that anybody that I'm working with right now has actually analyzed. Because we don't have that separate dispersant that has been analyzed using the same methods, it's a little disingenuous to say, oh, it's very easy to separate those two out. Because in the end, both were hitting the shoreline, and both could have consequences.
Thank you. I'm Charlie Pettit with Science News and freelance for Dr. Beasley. Correct me if I'm wrong, but I seem to recall just from lay press that in Prince William Sound, for some years later, you could pick up the rocks from the tidal flats and find a good deal of oil.
Yes.
Could you tell if this oil was cleared by October following this spill? Is the difference the weight of the crude or the temperature, or is there any way to sort that out? And second, I don't know if you just were looking in shallow sediments or if you dug down feet. If you could define a bit more how thoroughly you looked for some of these hydrocarbons, that would be great.
Right. Well, as far as the how deep we went, originally during June, July, and September of last year, we were going primarily just down 10 centimeters. That's where most of the oil we were finding was. However, in this past June, a year later from our original sampling time, we took deeper course because I was concerned that possibly it was being buried, because we've seen that, say, on some of the beaches, that the oil over time with the tides coming in becomes buried. And the problem is in the marsh, you can't see the oil because you have the grass, the sediment itself is very dark, and so whereas on the beaches you can very well see that delineation of where the oil is if you dig down. And so we wanted to know if in fact some of the oil had been buried, and so we went down further, approximately 30 centimeters, and within that zone we did not see any detectable oil, and that was measuring it with total petroleum hydrocarbons. Now that's not to say if other methods had been used, say the GCMS spec or some of the more technical, like for the PAHs, which tend to stick around a little bit longer. However, back to the comparison to the Prince William Sound, that was a very different system. And yes, temperature is a major factor. That is a subarctic area, and it's a fairly pristine environment. And so the bacteria that live in that sediment were not accustomed to hydrocarbons. Because, like I said, it was very pristine. It was subarctic. You have a different, at that time, the type of analysis that we have now was not available at that time, but the type of bacteria that can live in that particular system is going to be very different or behave differently than the ones that live in our system. And so it was, yes, factors of temperature. Also, I believe you were saying the rocks and so forth. This is not a rocky area. This is primarily sandy, loamy marsh system. The temperature at this point, it was mid-July, August of last year, and so as we know in southern Alabama, the temperatures there are 90 to 100 degrees C, or 100 degrees Fahrenheit, and the water itself was approximately 30 degrees C, which is much warmer than what you saw at the Prince William Sound. And also, as I was saying, the Gulf of Mexico is one big hydrocarbon seep in itself. And so it has been shown in many studies that the organisms that live in these systems are very accustomed to this oil or to any type of oil. And so if the hydrocarbon degraders are there and you give them a natural carbon source, such as a hydrocarbon, they're going to take advantage of that.
And our third presenter, who wasn't able to be here, is going to call in at some point. Are you there, Peter? Anybody? All right, maybe not yet. But in the meantime, a couple more questions.
Thanks. Two quick questions. First, were you surprised that the bacteria basically cleared the area of oil in just a couple of months? And sorry, go ahead.
No, go ahead. Go ahead.
And secondly, has anyone considered figuring out which bacteria are doing the job, isolating them, and using them for future oil spills to help clear up oil?
Well, as far as being surprised, I went at this not necessarily with any preconceived expectations one way or the other. Because of the fact that the oil really at the beginning never was as high as some of the other areas along the Gulf Coast that were affected. As I said, in Alabama, for the cleanup standard is 100 milligrams per kilogram. So any type of a below ground tank that is leaking, if it has TPH, total petroleum hydrocarbons of 100 milligrams per kilogram or greater, it has to be cleaned up. And so we were only just a little bit above that, approximately 189 milligrams per kilogram was the most that we were seeing. And what was very interesting that I found was, where we found that it was so heterogeneous in the system that you could have a sample here and it would have no detectable hydrocarbons, but right next to it, it would have a very high, up to a high concentration for what we were measuring. And so, but what we saw also was that along those marsh grasses, like I was saying, the marsh grasses themselves seemed to be collecting the oil and allowing it to be distributed in the sediment, and that's where we saw the most of it. And so that was where the most, many of the hydrocarbon degraders we saw. Now, as far as the culturing and so forth, there is a long history or a long body of work out there on the bacteria that can degrade hydrocarbons. And what we were seeing in our system were many of these very same ones, particularly the gamma proteobacteria. These are very, aggressive type hydrocarbon degraders. And tomorrow I'll be a little bit more specific about some of the ones that we found. And some of these have been cultured in laboratories. We do not have at this point any plans to do that, but what can be interesting with this phylochip that we're using, this is the first time that this microarray analysis has been done on a marsh system such as this. And when it comes to hydrocarbon degraders in the past, you have to, being able to isolate them and being able to grow them and determine if they are hydrocarbon degraders. And so we have a certain body of knowledge that we know which ones can do this. However, with this particular type of analysis showing so many different bacteria that are actually there, we might be able to say, oh, we see these increasing in abundance. Maybe we need to look at these. People in the past might not have realized that they are hydrocarbon degraders. So.
And I think that beep. Yep. Peter, would you like to start?
Peter, I'm just stepping in. Sorry, I'm here.
No worries. Do you want to start your presentation and just kind of walk us through what you found?
Sure. Is this an appropriate time?
This is great.
Okay. Well, sorry you won't have any visuals, but my poster will be available tomorrow afternoon. Essentially what we're doing is we're looking at several molluscan or shellfish species to look at the incorporation of components from the oil spill, specifically trace metals and any PAHs or polycyclic hydrocarbons into the shells and the tissues of these animals. And eventually what we'd like to do is to use these different molluscan species to model possible pathways of distribution of these materials into the coastal food web of the Gulf. What we've done so far is to focus on the American oyster, Crassostrea virginica, partly because it has such a robust shell, and we understand the growth and geochemical system quite well, and also because of its obvious economic importance on the East Coast and in the Gulf. So we've been looking at trace metals in the shell, trace metals in the tissues, and we've also been looking for possible effects on tissues within the animals of the exposure to the oil. One of our concerns when we started the work is the fact that the Gulf itself is a naturally hydrocarbon-rich environment, both because of natural seeps and so on, as well as the petroleum industry. Since the 1930s, we've had so much production in that area. So our first goal was to establish a historical baseline of what contamination might look like in these animals without the Deepwater Horizon event. So we've been sampling specimens dating back to the late 19th century from museum collections across the country. We've also been looking at then specimens outside of the Gulf of Mexico, but also within the area of the spill, we have collections of shells from May 2010, before the spill made landfall, and specimens collected then in August 2010 that would have been exposed to the oil for a full summer, and then December 2010 also. And basically what we have right now is within the shells, the shell material of the oyster itself, we have in what we call post-spill specimens from August 2010, significantly higher levels of vanadium, chromium, and cobalt, which are all potentially sourced from the oil, in the post-spill specimens compared to specimens from the 20th century as well as May 2010. Similarly, inside in the tissues, gill and muscle tissues, we have significantly greater concentrations of vanadium, cobalt, and lead compared to tissues from specimens collected in May 2010. And these are all from the Barataria Bay area of Louisiana. And then finally, we've been doing histological analyses of the gill tissues. The gill is a very delicate structure in these oysters. And normally, the gill tissue consists of something we call columnar epithelial cells, where the surface cells take on an upright column-shaped configuration that maximizes the surface area for gas exchange, but under certain conditions of disturbance, chemical mutagenesis, and so on, that will be converted to a layered or stratified configuration that's called metaplasia. And we have evidence of that beginning in Barataria Bay in August 2010. Then we also have specimens from December 2010 from Ecological Bay in Florida. And all of those specimens show alteration of the gill tissues, or this metaplasia, So at this point, what we have are indications that trace metals that are present in crude oils, that's vanadium, cobalt, chromium, and lead, are present in higher concentrations in the shells or the tissues of the animals, and that the exposure to the oil may have altered the tissue structure of the gills of these animals. However, there are two caveats. The first one is we need many, many more controlled specimens, so we have to look at more pre-spill specimens and more specimens from outside the Gulf of Mexico to really confirm this. Right now it's statistically significant, but sample sizes are small. And also, we expected when we sampled the shells, we sampled in the direction of growth, and these shells grow like tree rings. You can read their histories. And so we expected to see an increase in these metal concentrations that would signify the onset of the spill effects, and we don't see that. Instead, the metals are distributed uniformly throughout the shell in these higher concentrations, and that signifies to us that the materials are being absorbed into the shell by a process we're not aware of, we don't understand yet. The other caveat also is that some of these metals are present not just in the oil, but in polluted fresh waters of which we had a lot flushed into the Barataria Bay area in an effort, of course, to push that spill offshore. So we can't say for sure that these metals are coming from the oil itself, but right now all indications are that the higher concentrations are a result of the Deepwater Horizon event. There. I'm sorry, this is non-video.
No worries. Thanks, Peter. Questions for any of our three speakers? I know we have one coming from the internet, but I'll hold off on that if anyone has any other thing to ask. Oh, well, I'll ask another one for Peter. This is from Seth Bornstein from the Associated Press.
Okay.
So, Peter, with the metals you found in the shells and the gill medical issues, how much harm is there? Are these fatal or not?
Yes, that's currently unknown. One of the interesting things is we don't know very much, if anything, about the effects of these metals on the oysters themselves. In humans, of course, we understand quite a bit about metals such as lead. We know that vanadium is present in the human body and at a certain point if the concentrations become high enough, there are neurological disorders. But how much of the concentration that we're finding in the oysters, which are very, very small, how much would be required to produce noticeable elevated concentrations in humans? That's something that we're not able to say yet, but we are interested, as I said at the outset of my talk there, in doing some modeling of the food web to see how these materials could become concentrated or bioaccumulated in like higher trophic level species such as large commercial fish that of course are feeding up the food chain from the oysters.
This is a question from Rich Monastersky from Nature. I just wanted to ask, there's been lots of other studies of the effects on the food web, and do you know of other results that show bioaccumulation of metals, either showing that they are or not getting into shellfish and fish and other organisms?
We do know a bit about bioaccumulation of mercury and lead. These are metals, because of their industrial applications, have been monitored for a very long time by the EPA and other agencies such as Mussel Watch from NOAA. So yes, we know that bioaccumulation can occur. And of course, with mercury, that's been a significant problem.
Oh, I'm sorry. He has a clear from the oil spill itself. That's what he meant. Oh, are there other studies showing similar things?
A similar thing is in bioaccumulation.
Have other people confirmed your results with other organisms or other samples from other places? That's what I'm asking.
Not that we're aware of, no.
So you're not aware of any other studies that have shown metals getting into organisms?
Metals from this oil spill getting into organisms?
Yes.
No, but we might become aware of some of those studies at the meeting this week.
Okay, thanks.
And I know we have one more clarification from the web. For Peter, you were talking about the gills. Can you describe the effect of gill alteration, and can they breathe still?
Yes, so I cannot say for certain what the impact of that transition from a columnar epithelium to a stratified one would be right now. That itself would have to be a separate study to measure the effects. It would have to be done experimentally. But given that the purpose of this columnar epithelium is to increase the surface area for respiration, when it's transformed to a stratified morphology, then you're decreasing that surface area, so it will have an effect on gas exchange. Gas exchange will be less efficient. How that translates into physiological downgrade, reproductive downgrade of the animal, that would require further experimental work.
And I have a question for Peter as well. This is Lizzie Grossman, freelance. Are you aware of anybody looking at trace metals in shrimp in the Gulf from the spill?
No, I'm not specifically. The whole, this idea of looking at the trace metals originating from oil, as far as I'm aware, there is one study that was done by a French group after the wreck of the tanker Erika several years ago, and they looked at the presence of metals in mussels on the shore, blue mussels, and they confirmed the presence of vanadium and nickel about 6 months after the spill. That's primarily where we derived our idea of going after the metals in these various molluscan species in the Gulf. But it is known, the presence of these metals is known in crude oils. There's a lot of work that's been done to characterize oils based on their metallic content, and I do know of at least one study, and I can't think of the investigator's name right now, at least one study that I think are looking at fish otoliths or air bones for the presence of metals.
Thanks. Any other questions? And with that, thanks so much to our speakers.
Thank you.
Sure. We'll have more press conferences tomorrow.